we investigated the effect of the plant beneficial rhizobacteria, or it's also called PGPR for short, on the innate immune system of sorghum plants. Now, PGPRs in certain cases have the ability to trigger an enhanced defense response within the plant. The bacteria interacts with the plant root system, set up a chemical communication system, and that leads to the ability of the plant to sense potential pathogens earlier and also to launch a more effective and stronger response against the attacking pathogen. That way the plant expends less energy towards defense and more energy can go to grain production. In nature, in the field, crop plants like sorghum cannot move away from environmental stress conditions. They have to cope with what they have at that point. So plants have an innate immune system, and this innate immune system allows them to survive and proliferate in the immediate environment. This innate immune system is quite often based on chemicals within the plant, and we can refer to them as a chemical cocktail that can be changed according to conditions. So when a sorghum plant is attacked by a pathogen like the Trichum subplunihalum, it turns on its innate immune system. But quite often that might not be sufficient, and the plant has to expend more energy to survive, and quite often the grain yield is then less, or the plant might even die in the process. So in our research we looked at ways and means to enhance the immune system of the plants so that a susceptible plant can become tolerant, a tolerant plant can become resistant, and eventually yielding more grain yields that can contribute to sustainable food production. Now, in this specific example, we used what is known as plant beneficial bacteria to form an interaction with the plant roots and thereby leading to a chemical communication system between the bacteria at the roots and the plant shoots that is encountering the pathogen attack. That chemical communication was then investigated by means of metabolomic tools and approaches using chemometric analysis.
In this study, we generate big data, gigabyte size, that are so complex. So we employ chemometrics, machine learning methods to extract information from such big data sets. So the information we extracted shows us that the primed plants change the chemical defense for a stronger and effective security system. So the changes that we observed included increase in the levels of lipids, increase in the level of amino acids such as tryptophan, and also the changes in flavonoids, which are chemicals that the plant uses to defend itself. So such flavonoids or phenolics.
So we made a significant amount of progress towards understanding the phenomenon of bacteria-induced priming and associated crop production. Our findings can also be extrapolated towards other crops and other pathogens under different environmental conditions.